Okay, now we're going to do testing for proteins, for the presence of proteins. And that's going to be with the Biurets test. So we're going to use this reagent, which has this beautiful sort of periwinkle blue color. Okay, so again, go through and you hypothesize. Do you think that distilled water will have protein in it? How about glucose, albumin, starch, or your food sample? Which of these do you think will have protein? Yes or no? And then up here it tells you this reagent, which is a blue reagent when it starts. If protein is present, it will turn this violet kind of a purple color. So if you're not sure about violet, just put down sort of a purple color. So if your sample goes from blue to violet, that means you're positive for having protein in that sample. So write what you think will happen here. Okay? And then what we do is we get 6 test tubes again. We label them, right? So we have water, glucose, albumin, starch, and French fry. And then it says put 5 drops of this Biurets reagent. Oh, I kind of did it backwards. So I did 3 first. I did this. And now I'm going to put 5 drops of these substances in here. So I'm going to start 5 drops of water in. 1, 2, 3, 4, 5. 5 of your glucose. Oops. Here's your albumin. Here's your starch. And the last is going to be my French fry. One, two, three, four, 5. Maybe not the most beautiful squirting. Sort of give a little shake here. I'm going to tilt the tubes up so you can actually see colors here. Maybe I'll even pull them out. So this is kind of a nice comparison because here we have, you can see, here's water. It still has that blue color. Here's glucose, also blue, but albumin, that's that violet color I was talking about. You can see how it's pretty purple. Kind of a nice color change. Here's starch. Again, that's pretty blue. And then the last one is your French fry. Also pretty much blue. Almost a little gray in color, but the overall idea here then, right, is you come down to this table, my camera to write what these final colors are. So water was blue, glucose was blue, starch was blue, but albumin was the purple. Unknown, we don't have, but the food sample also pretty much just blue. And though that means that this is a negative result for protein, so your conclusion would be no protein. Right? And then this one says what's your evidence? Your evidence is going to be the color of the sample. Okay?